integrated personal omics profiles. I always tell people like that this is a story of only one person, like in the next 15 minutes or so. I would like to tell you, like I know this person molecularly much better than even than himself. And first, of course, I'll start with some background. And the first background, of course, is technology development. And it's like the recent technology development in the past 2 decades especially has enabled many, many things, like especially large-scale profiling that people couldn't even think of before. For example, back in 2000, which was only 14 years ago, you could see that like at one time you could profile or genotype 600 SNPs, a single nucleotide polymorphism, which everyone has. That was considered high throughput because at that time, most people were looking at only one gene, and standard sequencing was only one sequence. But this number quickly rose to about 15,000 in only 3 years. And thanks to different companies who developed all these amazing sequencers, especially early this year, Illumina especially, and Oxford Nanopore, they claimed that they could do $1,000 genome, a US dollar genome, which completely brought down the cost for sequencing. Now you can sequence a single human genome at 30x coverage, we call depth of coverage, for about $1,400 US. Of course, there are developing other technology. For example, there are better mass spectrometers, which would enable people to profile more of the proteome and metabolome. And also some other platforms, like, for example, this was developed in Dr. Mike Snyder's laboratory, which is a protein microarray, which allow us to look for possible protein-protein interaction as well as the autoreactivity, like the autoreactive antibody profiles. On the other hand, in the meantime, I would like to bring up this molecular understanding of human health. It is like our health. Now we know that it's not only determined by ourselves, our own genome, our own body. It is actually also co-determined by the environment we live in, as well as the interaction between us and the environment. And it is true that since we have the first draft genome back in 2001, that we've been asking this question is how much can our genomic information can actually tell our health status? The situation, something happened here. Uh-oh. Okay, it's back. The situation might be simple for Mendelian traits, whereas the loss of function of one gene can directly lead to the phenotype. For example, a loss of function of a gene called PAH could lead to phenylketonuria. Loss of function of HTT can lead to Huntington's disease. For example, this is what we found last year, a new gene called TGC7A. A loss of function of that could lead to a severe phenotype called combined immune deficiency with the multiple intestinal atresia. So the formula, as I said, it was simple. It's a gene loss of function equals to disease. However, this gets more complicated when it gets to complex traits. For example, for height, in the genome-wide association studies catalog hosted at the NIH, you can see that there are more than 500 SNPs being associated to this trait. And this number was about 1,000 for another trait like obesity. And for some diseases, there are 218 SNPs being reported to be associated to the disease. But the problem is that neither a full set of these SNPs nor a subset of these SNPs has been shown to be sufficient to cause the phenotype. That's why let me borrow a quote from Dr. Stephen Saucer from the University of Cambridge. Like, these complex traits is more like Schrodinger's cat than Mendel's peas. And I think this is another, if Professors Luo and Zhang are here, this is another thing to weave biology with physics. And so in most cases, is because genomic information is not deterministic, it's actually probabilistic. And disease, this is my formula, is like genetic predisposition plus environment effect plus maybe some trigger. But the problem is environments, a lot of times, are really hard to quantify. We don't know what to quantify, what to measure, what to quantify, what to put in our model, for example. But my idea is that our response is not. We could capture our response. First, we could capture the genome information in us so that it could predispose us to different medical conditions. And then it is our interaction with the environment that can be reflected at different molecular levels, such as the epigenome, the transcriptome, the proteinome, the metalome, and so on and so forth. And it is in this background we carried out since 2010. I think it was like the craziest project I've taken on. It's called Integrated Personal Omics Profiling. The reason is that we're all made of molecules. And we termed it IPOP for short, Integrated Personal Omics Profiles, because we're all Apple fans. And the goal of our project is to try to attempt to obtain a detailed molecular portrait for health and disease. And the aims that we had was, first, we would like to identify what would be quantified, what could be tracked, and what would be quantifiable. And then we would like to see how they changed over time. And then in the end, we would attempt to interpret these omic types and link these omic types to phenotypes. And this is a concept hypothesis. It could be DNA, it could be modification, DNA markers, epigenomic markers, or we add, like on another layer, we add RNA molecules, profile of RNA, profile of protein, profiles of metabolites, and we add all these together and makes a complicated, I call it a kind of a 3D snapshot or multiple dimensional snapshot of this person. And hopefully, when we track this snapshot a long time, we could see the changes in the snapshot that actually could reflect that person's actual physiological status at the time the sample is taken. This is, we call the test subject. We worked on this one person who was 54 at the start of the study, who works at Stanford, of course, is a Caucasian male with the normal BMI. And he was generally healthy at the start of our study. And we have followed him for more than 4.6 years. And we passed the 4-year mark in this past March 29. And today, I would like to share with you some highlights, and just a few highlights because of the time, that what we published in the first 400 days, which contains 3 biological events. A human rhinovirus infection, that's a common cold viral infection. A RSV infection, respiratory syncytial virus infection, which is also a common cold viral infection. And third, right after RSV infection, the early onset of type 2 diabetes. And so I would jump straight to, skip all the genomic part and jump straight to integrative omics. Is because, as I mentioned before, the genomic information alone is not sufficient to tell that person's real-time health status. That's why that moves us down to more dynamic molecular levels so that we could potentially use these to reflect that person's health. And we generate terabytes of data from this person. And we both look at these specific, separate data sets separately, and also put all these together and look for mainly 2 features for our study. One is autocorrelated features, meaning that these every time point is related to the previous time point, the status of the previous time points, and which also could, for biologists, I could explain it as something like trend. And the other one is spikes, meaning like a significant positive or negative spikes, which I could interpret as peaks. And here is a summary of all our significant data sets as a result that we found. And I would like to just follow a very short story with you here. So I first started by profiling that person's cytokine levels in his serum. You see that at the start of both infections, there was a significant upregulation of pro-inflammatory and anti-inflammatory cytokines. But there was a third peak upregulation of cytokines that was not accompanied by upregulation of C-reactive proteins, which is a known acute immune response protein. And but interestingly, right after that, his fasting glucose went up high. And during the course of study, he was officially diagnosed as having type 2 diabetes. So our question is whether we could see these corresponding physiological status changes in our omics data. And for example, this is the integrated changes that we did. And we could, using unsupervised hierarchical clustering, we could break these into these four major clusters. One of these clusters was enriched in pathways related to both immune response and cell proliferation, which symbolizing the body's response to the infection. And there was another big cluster, was later the peak being later on day 18, which were enriched in pathways related to metabolism, like glucose regulation, insulin secretion, like pyruvate metabolism, TCA cycle, or metabolism of carbohydrates, as well as some diseases. So like Huntington's disease, Parkinson's disease, Alzheimer's disease. So I was a little bit curious, and so I searched on the PubMed. Actually, when I searched diabetes and Huntington's disease, there were more than 200 articles reporting that. And when I was doing diabetes and Parkinson's, there were more than 1,000. And the diabetes, Alzheimer's, there's almost 3,000 articles there reporting the potential association. And actually, this specific group is even trying to promote, to call diabetes, type 2 diabetes, as, Alzheimer's disease as type 3 diabetes. And of course, we also looked at identifying many other changes, like the negative changes, as well as autocorrelated changes, as I mentioned earlier. And in the autocorrelated changes, you can see right after the RC infection, there was a major cluster was gradually going up, which are enriched in pathways, biological processes that have the term metabolic process or catabolic process, as well as another major cluster was gradually going down, which were immune response, which actually indicates that at that time, that person's infection was gradually clearing up while his symptoms of diabetes was kicking up. And here's a just short summary of that, which has contains all the data I didn't have time to show you today. That at both infections, day 0, HRV infection was a peak of cytokines, C-reactive protein, and transcriptome. And for RSV, there were more spread. You could see the peak of cytokines showed up on day 0, and 1 day later was transcriptome, and followed by C-reactive protein, and proteome happened last. And they were enriched in immune response related signatures. And on type 2 diabetes, on day 12 post RSV infection was a peak of cytokines, glucose, as well as integrated metabolome. But 6 days later were the peak of transcriptome, proteome, as well as the integrated profiles. And they were enriched in metabolic process related signatures. So the conclusion is we did observe IPOP signatures that were concurrent with the happening of his physiological status changes. And one question we asked ourselves is that does integrated omics, and also other people asked, whether the integrated omics could tell us more information than we looking at each organism separately. Thank you. And this is in our own data. It's not so new that RNA and protein expression levels do not correlate well, but actually we were still surprised when we saw that in our own data. It is true that some of the enriched pathways that we identified were made of major from of RNA components, and some were mainly of protein components, and some were really mixed. So the answer we found is yes. We look at all these, so they only partially overlap with each other. They actually add complementary information on top of each other towards a whole full understanding of that person's physiological status and status changes. So the conclusion is, by generating so much data on this one person, we did profile that person's integrated omics and completed the principal study. We estimated that person's genomic risk, and also showed that integrated personal omics could provide a specific perspective towards understanding of that person's of any physiological status and status changes. The directions is some of the vision, because after I'm still in the study, it's still ongoing, but after we published this, I've been thinking what we learn from this, and in the future, what's the future, like the next step of this. Eventually, I would envision we need to change these molecular snapshots to molecular symptoms, so that we could achieve personalized health monitoring and preventative medicine. And here I would like to emphasize the power of N=1 is that because many of these previous studies are done on a large population, the case population, control population, but they didn't consider interpersonal differences. And sometimes these differences are too huge so that it could give significant signal that change in one person, but will be lost when you pursue these in two very general groups. And then I'm a deep believer at these non-hypothesis-driven or data-driven modeling or studies would feed back to hypothesis-driven studies, and all of these would go into a very productive loop driven by curiosity and beauty that the two professors mentioned before. Of course, it's still unknown that what's the resolution of omics, right, like required that we need for these omics profiling. How many ohms do we actually need to capture, let's say, 99% of health-related information? And whether we should keep everything, or we should actually reduce this to a small set, or handle the set, which is a hot debate of holism versus reductionism. I'm more towards holism by the way. And also, if we could achieve personalized medicine, who might be responsible to synthesize, to develop all these personalized drugs? But hopefully, before I die, I could see that any average person goes to the clinic, that person already have these multi-molecular profiles stored with the doctor, or even with themselves, and with all these molecular profiles being measured, constantly measured, and it could help with the risk prediction, and whether something goes wrong, like it was early detection, and if something goes wrong, early diagnosis, and personalized treatment. So here I would like to stop and thank my postdoctoral advisor, Dr. Michael Snyder, for giving me a chance to work on this crazy project, and also my colleagues in the IPOP team, especially Drs. George Mias, Jennifer Li Tan, and Li Huajiang. And I would like to share with you this picture of Dr. Mike Snyder here, is that I would like to thank him as the test volunteer of our study. And finally, yeah, you can see that he's holding a flask in his hand, and there was a little him inside. It's a personalized medicine. And last of all, I would like to share with you this picture. I was shocked when I saw this past January. This is taken by the Curiosity rover on Mars. It just looked back and took a picture. It's too hard to see. Of us across the Earth and the Moon, you can see from far away we're no difference from the Moon. Only if you zoom in to the planet level that we see. You see that, right? Like this is.
It doesn't matter. It's art. It's okay.
You get the Now you get it because it's no difference. That's like an interpersonal, right? Differences. We don't really see that until we really look into these details. And many thanks for your attention, and I'll be happy to take questions. Thank you.
Do you have questions here? Okay, so I think people prefer to have a. Okay.
Yes. We can talk offline if you want. Please, sorry.
Private talk.
Sorry. So my question is very kind of a general one. You have demonstrated that using molecular profiling, very large scale molecular profiling, you can kind of correlate the profile and the physiological state and predict the disease risk. And my question is, what's the advantage of this very large scale molecular profiling over the very simple diagnosis test, like blood test?
That's a great question. That's actually in line with the debate I listed before, the holism versus reductionism. Many people actually have argued that, especially for type 2 diabetes, if you can diagnose with glucose, why would you still need to study that, right? Like, and that if somebody has type 2 diabetes, just give them insulin shots, ask them to exercise. But in fact, there are many, like, I don't know, thousands of at least researchers in the world still working on diabetes. It's that we still don't know the molecular mechanism. Meaning that knowing that only one molecule change is not enough for us to know the mechanism behind what's actually happening. And this large-scale profiling will tell us a full story of what's actually happening molecularly in our body. And it might not just be, for type 2 diabetes, it might be easy, right? What if there's some disease that was undiagnosed, that nobody knows where to look at? And this can tell us the changes in the body of the patients over time that can tell the information both related to that disease and the information related to other physiological changes. I do believe this is more like a 3D hologram type of picture of that person, and that can help us understand more of ourselves, how actually we operate on our, how our body operates on itself, right?
Okay, more questions?
Yes.
Okay, good. Let's thank him again.
Thank you.
Second invited talk will be given by Dr. Yujiao, and she's a female representative of our female postdoctoral community in Stanford. So just now I heard too many talks from the guys, so maybe it's good to have a girl. So Dr. Sun is a postdoc from UCSF, and she has a particular research interest in the development and wiring mechanism of the neural network. And one of her study in the auditory critical development was published in Nature. So let's welcome Dr. Sun.
Thank you. Thanks for coming and giving me this opportunity to present. So I recently came to Dr. Roger Nicoll's lab at UCSF. So it's been a year, but I don't have enough data to show you the whole story. So actually I'm here to present my previous data and some undergoing manuscripts. I'm still working with my graduate PI, Dr. Li Zhang at USC. So I want to talk about how this auditory perception are emerged and developed, and also what is the underlying circuitry mechanism over there. So by starting, I want to concentrate on this particular word, circuit. So this is probably one of the most talked words in this Bay Area, the technique area. So the circuit is defined as an assembly of components work together to carry out certain functions like filter the information and integrate and also carry out the decision according to a preset algorithm. And so far, the most complicated circuit to us is actually our human brain, so which contains this, Dr. Luo already mentioned, trillions of neurons interconnected with tens of trillions of synapses. And then that could enable us to perceive and think and calculate and learn and memory and even change our behavior to adapt to the environment. So these are just some factors which are like, we got 20 billions of neurons in only in the cortex in human being, and then multiplied by the synapses for each neuron, which gives out the 0.15 quadrillion synapses. And if we think about how many of those nerve fibers are there, it will give us to 150 to 180K kilometers. So that's a lot of them, and it looks like a mass. And even between the hemisphere in the same person, we can see that there's 186 billion different neurons, which is like on the left hemisphere, we got more than the right side. So this is such a complexity and structurally challenging object. So it seems like at the first sight, it's impossible to ever decipher this black box. But if we look at it closer, we can see that among all those superficial disorganization, we can see there's some repetitive patterns and some stereotypical organizations that we can actually follow. So for example, among the different cortical area, like motor cortexes, sensory cortexes, or the frontal cortexes, they all share this laminal structure, which have the 6 different layers of neurons, and also each layer contains a certain type of neurons and have their stereotypical shape. So probably that's underlying something that maybe this specific structure could give rise to their particular functions. And also, if we check out the circuit, if we check out the cortex, and we can see that there's actually some beautiful stuff over there, the simplicity. Like if we look at the sensory cortexes, they actually have this famous homunculus, which is the, although disproportionately, but still in a spatially ordered presentation of our sensor, so that from toe to nose, we can see that that represents the whole body. And in the auditory cortex, which is the area of my research interest, you can see that it's the primary auditory cortex as this beautifully lined frequency retinotopic map, which is like the neurons can be lined according to their purport responding frequencies. And also in the hippocampus, you can see that they have all these great cells where they can show all the GPS maps. And actually, if you look at them between those different sites, the distance from each type is 1.42, which is the square root of the number 2. So this is actually like our camera, which we get the zooming out the this particular ratio. So from all those structural underlying, that probably we can see that there is a way for us to dissect all the mysterious that brain carries that allow our behavior and giving us an opportunity to explain all this using a synaptic and circuitry approach. So, for example, when we look at the simple behavior of reading a book, there will be several pathways involved in it, which is like the sensory cortex allows us to perceive the words in the book, and then that can send out to the frontal cortex, which allow the perception of the meaning, and finally there will be the motor information that goes back there to allow the eye movement as we scan the book. And within this pathway, there will be the little information center where it's called the local circuit. The information can be processed locally, both horizontally and vertically along the laminal organization. And from all this local circuit and pathways, this single neuron will be the unit that can actually carry out the function as the independent electric component, which the neuron will integrate all sorts of different inputs and then generate the electrical impulses and send it to the next group of neurons. And if we look closely through the resistor that would allow the flow of the ion channel in and out of the circuit. And so based on this simple simplification, so in 1952, Hudson and Huxley came up this famous model to interpret the electrical features of the neuron. So basically, all they did is calculate the current, considering all different electrical properties of the neuron. And then, although this is a mathematically unsolvable equation, but still using the simulation, we can very faithfully regenerate this spike tuning curve of the single neuron or the axon, as they showed it. And this work got the Nobel Prize in the 60s for this great description, the simple and beautiful explanation of the biological term with the mathematical modeling. And also, one thing I want you to take a look at this, the nonlinear property of our neuron, which is like when the neuron integrates all the input, it will make a decision. It's not simply just give a readout, but only it will decide whether this integrated response exceeds this threshold level, where once it hit there, the neuron will generate a spike. Otherwise, it will just let it go instead of making the output response to the next stage. So even with all this great property, we wouldn't be able to actually tackle the question of this electric circuit without this advancement of electrophysiology, which is developed by Avenue and Berstachman, which show that using a glass pipette or an electrode, you can actually approach the neuron and suck up part of the membrane, and then measuring this the change of the voltage differences, so that you can actually get the output response of the neuron. And moreover, if you actually break the membrane and use this glass pipette to follow the whole circuit with the membrane, you can actually see, you can actually do a readout of the intracellular responses. So by doing this, you can block certain type of ion channels and then give you all the readouts
with the neuron spikes as well as the subthreshold responses. And if you do it more aggressively by holding the neuron at certain voltage, you can actually create a reverse potential for the rest of the ions, and then just get the ion flows of your own interest. So basically, with all this great technique, we were able to tackle the circuitry question with the single cell patch clamp. So either you can use it to get the readout, or you can see that with the voltage clamp technique, you can see all the excitatory and inhibitory input targeting to the specific neuron. So with all this, my question will be, what's the circuitry mechanism underlying this emergence and development of the auditory perception? So just a quick review of the auditory system. The sound is actually the mechanic wave, which is converted to the electric signals by the hair cells in the cochlea, and that will send the information up to the central auditory system, all the way up to the auditory cortex, which is a stage for perception. And if we look at the auditory receptor field, instead of the spatial receptor field in other sensory systems, actually the auditory receptor field have this specific response to the frequency, to the sound frequency instead of the spatial location. And they have a typical V shape, which show that they can respond to sounds at certain level. For each single neuron, they can respond to sounds only to certain level, and also with their preferred frequency. So the way we do the experiment is to give the different subtonal stimulation, and then using the electrophysiology to measure different responses. And afterwards, we can align those responses to different frequency and intensity to create this whole map. And also, by measuring each amplitude of by measuring the amplitude of each response, we can derive this beautiful tonotopic receptor field of each single neuron. So, as I already mentioned, there is a tonotopic map, and also there is a laminar organization, which shows that particularly in layer 4, that is the recipient layer, which receives the subcortical input and then integrates and then send it to other layers for further processing. So our very first question will be, how does sound perception ever emergence? Does it require the nurture, or it's simply already defined by the nature? So by recording the neurons at P12, which is the day the ear canal first opens, so that the cells can sense the mechanical wave, we already see that there is a very pretty balanced match, the excitation and inhibition. Since the inhibition is only in the cortical area, it's very local. So that actually means that the descending pathway as well as the local circuit is already there even before any external stimulation. So our finding first define that the nature already shapes this pathway, probably due to some genetic coding, or maybe the internal spontaneous electrical waves within the circuit. And then we also want to see whether nurture play any roles, because as in other systems like neuromuscular junction or the visual system, they already show that they do require the activity to do the pruning and get this refinement of the responses. And also skip this part, but basically we already see that there's a refinement along the stage. So using the electrophysiology, we can further dissect the excitatory and inhibitory circuits, and seeing that along this, and find that along this development, the activity actually mainly refines the excitation, but not the inhibition. So furthermore, by doing the integration using a simple integrated fire model, we can recapitulate this the neuron output refinement along the stage. So in conclusion, our work showed that the nature determine the pathway, which means there's a balanced excitation and inhibition. And then the nurture at the later stage helps sharpen the responses. So the following project is to understand what part of the excitation that's actually being refined along the development. Because we can see that as the recipient neuron, as the layer 4 neurons in the recipient layer, they receive both the thalamic input and the intracortical input. So the refinement could be just due to the thalamic output, or because of this projection, or even it's just locally at the cortical area. So first, by measuring the thalamic responses, we can see that although there's a refinement of the tonotopic mass, there's no much change of the neuron responses. And then we balance the intracortical activity and review that the thalamic input along the development actually already got refined. And that's also the case for the intracortical input, which is derived by a subtraction of those thalamic input of the total responses. So in this second part of the work, we further demonstrate that this refined thalamocortical input is the major driving force for this excitatory refinement. So in conclusion, my work shows that this nature and nurture shape the excitatory circuit altogether, where the nature first defines a pre-wired structure, and then the nurture can further sharpen the auditory responses. And the underlying circuit is mainly by this excitatory contribution by the thalamocortical and intracortical input. Finally, I'd like to give thanks to my graduate PI, Dr. Yujiao and Rui Chen. And thanks for your attention. Okay. Two invited speakers for the inspiring talk. So do we have questions here? Okay, so if you take the time of the coffee break, you can contact in person with Dr. Sun about any questions you may have. But before the coffee break, we need to invite all our silver sponsors here. They will say a word or two about their company, so please come in front in here. And afterwards, we'll have a 15 minutes coffee break. During the coffee break, please, the poster presenters, if you didn't give us your posters, please give us. We will have people in the opposite room. We'll have to just set the posters up. And also, can you all come together? Thank you. And also, please do come back, because at the very beginning of the next part, the panel discussion, we will have another raffle tickets drawing. So if you leave in the coffee break, then you will miss the chance. So. Okay, so let's start our sponsor talk.
Thank you. I'm from BGI Tech. BGI Tech is a service division of BGI Group, Huadaji, and our U.S. branch currently has 2 sequencing labs, one in Sacramento, another at Philadelphia, and with this local support, and so we are proud of serving some of the Stanford labs for years till now. And our service line has all the different kinds of next-generation sequencing services, including transcriptome sequencing, resequencing, and methylation sequencing, and single cell, all the different things. And so come to my tables across the hall. And by the way, we are hiring. Thanks.
Hi, good afternoon. Thank you for the chance to present. My name is Jiangwen Majedi. I can really relate to you guys in Chinese. Jiangwen, 长江的江，文化的文。Majedi is an Indian name. Not many people have this last name, but there is one on this campus, and I'm related to him. I think it's next building. But anyway, a few words about my company, BioZero. So it is a CRO company. It's located in China, both in Beijing and Shanghai. But it is a relatively unique company because we belong to a global US company called PPD. PPD stands for Pharmaceutical Product Development Company. It's one of the top 3 clinical trial service companies. But BioZero itself, being a US company located in China, has many advantages. And it is focused mostly on preclinical research, drug discovery research. Particularly to any of you who are interested in starting a virtual company, startup companies, we're very good at helping startup companies to take off. Anywhere from drug discovery all the way to R&D. What we do is we have chemistry, compound synthesis, scale-up, medicinal chemistry, a lot of it. And then we also have a discovery biology group. We do reagent generation, and we also do assay development. And then other than that, we have a big Bavarian. We do a lot of ADME and pharmacokinetics work if you want to test the property of your drug in vivo. And then we have also pharmacology group. We do a lot of animal models. Most major areas include type 2 diabetes and inflammation area. And then we do also some safety exploratory talks. So anyway, for biotech companies, we do the integrated service, meaning we do all 4 areas, but we also work with academic labs. We do individual fee-for-service projects as well. So please, if you're interested, come talk to me next door. Thank you.
Hi everyone, I'm Seth Rubin from the company Active Motif. We're based in Southern California in Carlsbad, just north of San Diego. And I'd like to thank the organizers for inviting us to participate today. And incidentally, one of our newest locations in the world is in Shanghai, and Zhentian comes from that location. So thanks very much for inviting us here to participate today. We're happy to contribute, and we'd like to congratulate your organization on this fantastic program today. So thanks very much for including us. Just a bit about Active Motif. We focus on gene regulation and epigenetics, and we offer a full line of products and custom services for looking at epigenetics and gene regulation in terms of products like antibodies, recombinant proteins, assays, and like I said, custom services for looking at pretty much any area related to gene regulation and epigenetics. So if you'd like more information or a t-shirt, please visit me next door.
Good afternoon, everyone. Thank you all for staying along here. Our company is Christopher Huang CPC. We have 3 locations in the Bay Area, one in Millbrae, one in San Jose, and another one in Dublin. We also have an office in Seattle, Washington, and 2 consulting offices in China, Suzhou. And our boss, Chris Huang, has been specialized in certifying accounting services and tax regulations for like over 10 years. And he's been doing all kinds of accounting services and accounting company types, like pharmaceutical, real estate, financials, and startup companies, and public companies. And we also do international companies and the interrelationship among the foreign companies and the domestic companies. And if you guys have any questions or interests, I'll be at next door. Thank you very much.
Hello everybody, my name is Derek. First of all, I appreciate the ACSSS offering this opportunity to introduce my company. My company is located in the name is Try Bioscience. It's located in Colorado, very close to Stanford. The company focuses on development of novel biochemical assay kits and reagents. We have some multiple and diversity the assay kits to include the biochemical reagents and kits. This includes apoptosis, cell viability, ATP and ADP detection, and other mode detection kits. In cell biology section, we include the specific cell qualified FBS and IHS, the medium, and also other detection kits. We also have the protein assay kit and ELISA kit. So this focuses on the complement and the subkinds and some of the biomarkers. Next one is the macrophage depletion. So we have some very powerful collagenase bases, the Nexcelom. So this use a temporary deplete so the macrophage. The next one is viral vectors. So we developed some of the viral vectors for the research use. We also provide the customer service. So this is regularly used in the lab. As the founder, we have the lab and bees and some animal models. The last one is collaboration of platform. So if you have any idea or do something, so we can help you, we can support you. This is our company's the global information. If you need any help or need you have any question, you can contact us. So the address is up there, and also we have the phone and not here, but the website. So the address is here. And now the big news. So to support the ACSSS activity, so we offer the 20% discount for all ACSS members, and also we contribute additional 10% to assess the activity. This is the big promotion. So thank you very much.